Imagine a world where tuberculosis was a death sentence. This was the grim reality before the discovery of streptomycin. In the 1940s, a team led by Selman Waksman at Rutgers University embarked on a relentless quest to find antibiotics. Their breakthrough came in 1943 with the discovery of streptomycin. This powerful antibiotic was the first effective treatment against tuberculosis, a disease that had claimed countless lives. The discovery marked a significant turning point in medical history, giving hope to millions and transforming the landscape of infectious disease treatment. Streptomycin's impact cannot be overstated. It saved lives and ushered in a new era of antibiotics. Imagine undergoing surgery with no relief from agonizing pain. This was the grim reality until the mid-19th century. Enter Horace Wells, a dentist who, in 1844, witnessed a demonstration of nitrous oxide, commonly known as laughing gas. Intrigued by its potential, he tested it on himself during a dental extraction and felt no pain. This marked the dawn of anesthesia in medical procedures. Wells' breakthrough was initially met with skepticism, but it paved the way for modern anesthesia, revolutionizing pain management in surgery. Today, nitrous oxide remains a staple in medical practice, thanks to Wells' daring discovery. Imagine the early 19th century, a time when doctors had to press their ears directly against a patient's chest to diagnose heart and lung conditions. Enter Dr. René Laennec, a French physician, who in 1816 faced a critical challenge, examining a young woman with heart disease. Modesty and accuracy were at stake. In a moment of genius, he rolled up a piece of paper into a tube and discovered it amplified the chest sounds. This simple yet groundbreaking invention led to the development of the stethoscope, revolutionizing diagnostic medicine. The stethoscope, now a symbol of the medical profession, transformed patient care and paved the way for modern auscultation techniques. Ever wondered how doctors quickly assess a newborn's health? In 1952, Dr. Virginia Apgar revolutionized this critical moment. Facing high infant mortality rates, she developed a scoring system that evaluates 5 simple criteria: appearance, pulse, activity, and respiration, known as the Apgar score. This quick 1-minute test provided immediate insights into a baby's health, guiding doctors in urgent care decisions. Before the Apgar score, there was no standardized method. Now, it's a universal practice. Dr. Apgar's brilliant yet straightforward innovation has saved countless lives, forever changing the landscape of neonatal care. Imagine a world gripped by fear, where children everywhere were at risk of paralysis from a mysterious virus. This was the reality before the discovery of the polio vaccine. In the early 1950s, Dr. Jonas Salk developed the first effective polio vaccine, a monumental breakthrough that revolutionized global health. Before this, polio outbreaks were devastating, paralyzing, or killing thousands. Salk's vaccine underwent rigorous trials, proving its efficacy and safety. By 1955, mass vaccination campaigns began, dramatically reducing polio cases worldwide. This pivotal moment not only saved countless lives, but also exemplified the power of medical research, paving the way for future vaccines and global health initiatives. Ever wondered how we ensure the effectiveness of new medicines? In 1950, a revolutionary method emerged, the double-blind study. Before this, biases often skewed results, leading to unreliable data. In a double-blind study, neither the participants nor the researchers know who receives the treatment or placebo. This groundbreaking approach was pioneered by renowned British biostatistician Sir Austin Bradford Hill during the tuberculosis streptomycin trials. His innovation ensured unbiased results, setting a new gold standard in clinical trials. The double-blind study transformed medical research, ensuring more accurate, reliable outcomes, and remains essential in developing new treatments today. Imagine a world where diabetes was a death sentence. That was reality until 1921, when Dr. Frederick Banting and Charles Best changed the course of medical history. Driven by determination, they embarked on a groundbreaking journey, isolating insulin from a dog's pancreas. Their relentless work at the University of Toronto led to the purification of insulin, transforming it from theory to lifesaver. On January 11, 1922, 14-year-old Leonard Thompson became the first human to receive insulin, dramatically improving his condition. This monumental discovery not only saved countless lives, but also laid the foundation for modern diabetes management. Imagine undergoing surgery fully conscious and feeling every excruciating cut. This was the grim reality before Dr. Crawford Long's revolutionary discovery. On March 30th, 1842, in Jefferson, Georgia, Dr. Long administered ether to James Venable, a young man needing the removal of 2 neck tumors. Miraculously, Venable felt no pain during the procedure, marking the first successful use of ether anesthesia in surgery. This groundbreaking event paved the way for modern anesthesia, transforming painful operations into humane procedures and revolutionizing the field of medicine forever. Dr. Long's innovation remains a cornerstone of surgical practice today. Did you know that the first successful organ transplant was a kidney transplant performed in 1954? In an era where organ failure often meant certain death, Dr. Joseph Murray and his team at Peter Bent Brigham Hospital in Boston achieved the unthinkable. The groundbreaking surgery took place on December 23, 1954, between identical twins Ronald and Richard Herrick. Ronald donated one of his kidneys to his brother, Richard, who was suffering from chronic kidney failure. This revolutionary procedure proved that organ transplantation was possible, paving the way for future advancements that have since saved countless lives worldwide. Imagine the courage and hope that filled that operating room. Imagine a world where surgeries almost always led to deadly infections. This was the grim reality until Joseph Lister, a British surgeon, stepped in. In the 1860s, Lister observed Louis Pasteur's work on germs and pondered their role in post-operative infections. He began experimenting with carbolic acid as an antiseptic. In 1865, Lister treated a compound fracture with carbolic acid, and the patient recovered without infection. This groundbreaking success led to the widespread adoption of antiseptic techniques in surgeries worldwide. Lister's innovation drastically reduced surgical mortality rates, revolutionizing medical practices and saving countless lives.